So welcome to the photosynthesis webcast. In this case, we're going to be talking about going completely through photosynthesis. And as you can see, I already drew on the page for you a little electron transport chain, as well as down here at the bottom we have a chloroplast. So we can just write chloroplast there. And before we really get started, we just want to kind of go over the different parts to it. So if we are talking about the electron transport chain, we're talking about what's happening in this area inside of here. So we're going into the thylakoid here. And if we're in the electron transport chain, that would mean all of this area up above here is the thylakoid. So we're going through the thylakoid membrane into the thylakoid, and that's where we're going to be building up those hydrogen ions that we talked about before. So let's start at the beginning. And in this case, we'll start over here. We'll do the light reactions over here. And this is our photosystem 2. And don't forget, photosystem 2 comes before photosystem 1. And it does that simply because it was found first. Or photosystem 1 was found first and photosystem 2 was found second. So we have a photon of light that comes into photosystem 2, and that's just a chlorophyll molecule. Chlorophyll molecules are the light antennas, I guess we could call them. So the light is attached there, or kind of captured there, and we also wind up splitting water. So this is why we need water in photosynthesis. So here we have H2O, and we're going to split it to oxygen, and as we know, that oxygen goes out of the stomata of the leaf. So that shouldn't be anything new. So once that chlorophyll molecule gets excited, the electrons jump up, and they're all nice and high and excited, and then they go travel down this electron transport chain. And eventually, they kind of land in photosystem 1. So let's take this opportunity to talk about what happens as they go through here. If we look at our electrons come in, right, travel through here, right, and as they do that, all they are doing is pumping hydrogen ions into this space, and that's really what we want happening. So we have a whole bunch of hydrogen ions building up a hydrogen ion concentration inside the thylakoid. And the same thing that we saw happen with cellular respiration is going to happen here. These hydrogen ions are going to come through ATP synthase. We'll just write that on here for you. And once they come through ATP synthase, then we see ATP being made. And so we see ATP from ADP plus P. Okay. So we get out of this first electron transport chain, we are making ATP, and that's really what we want to make. That ATP is then going to travel over to eventually the Calvin cycle, which is the second step of photosynthesis. Going back to photosystem 1, when it gets here, we have another photon of light coming, and remember that they're not working against each other, they're working together. So it's a different wavelengths of lights that it picks up, and this happens to be a different chlorophyll molecule. This is actually chlorophyll B, and photosystem 1 was chlorophyll A. It travels down another small little electron transport chain, and eventually we get, let's use our green color here, NADPH, which is our electron carrier. So we have NAD plus, DP plus coming in, and we get NADPH, which also goes over to the Calvin cycle. So if we're drawing our pictures like we did in class the other day, what we see happening is, in this case, the electrons are full. And if we remember, this electron carrier up here is empty. So NADP plus is empty. NADPH has the electrons. Both of these are then going to go to the Calvin cycle. The second part of this, and this is called non-cyclic photophosphorylation. The second part of it is that we have to think about, well, what happens if there's no electron carrier for us? Well, if that is the case, then we need to kind of do something else. Once the electrons get to this point and they can't travel down this way because there's no NADP plus available, they kind of come back around and go back towards the first part of this. And when they do that, then they can travel back down, make a little bit more ATP. Awesome. And then once they get to photosystem 1, they can come back up again and try again. And this can happen again and again. This is called cyclic photophosphorylation because it's a cycle. Remember, that only happens if NADP plus is not available. So this is only if the electron carrier is not available. So we'll just write up there that there's no electron carrier available. And that is going to be our cyclic and non-cyclic photophosphorylation. We can put those down here. You should really be able to explain the difference between cyclic and non-cyclic photophosphorylation. If we look at this, cyclic means that it's a cycle. Photo comes from the light. I'm going to run out of room here, so we'll just kind of push it over down here. So this is cyclic photophosphorylation and also then non-cyclic. Okay. And that kind of covers our light reaction. So on this page we have the light reactions occurring. So they need light, and we already saw where that happens. So let's go to a new page, and let's kind of go through and do the Calvin cycle, since we said it was the Calvin cycle. So up here we'll write the Calvin cycle, and we'll go through it. And we know that the Calvin cycle comes in, it's a cycle, so we're going to have a nice little circle going on here. Right? And we're actually going to bring in 3 carbon dioxides. This is when our carbon dioxides come in. And there's 3 main parts to the Calvin cycle. The first part is going to be our carbon fixation, where we're going to take that carbon dioxide and we're actually going to fix the carbon into an organic molecule. And the organic molecule we're actually putting it in is RUBP. Okay. In order to do that, we need a couple of things. We're actually going to require 6 ATP. And we know we would get ADP out of it, and that ADP can then be formed again. We're also then going to go into our second step, and with the second step we're going to need some of our 6 NADPHs, and those are going to be reduced down to NADP plus. And we have over here our third step. And for our third step, we are regenerating RUBP for the second step. And that requires a little bit extra ATP, which is why we're really glad that we can actually do cyclic and non-cyclic photophosphorylation, because we need 3 additional ATPs here for this step. So what do we get out of here? We actually get a G3P, which will become our sugar molecule. We make 6 of them. We get to lose 1 of them. So 1 leaves the cycle. And there are 5 other G3Ps that have to become, or kind of become rearranged into RUBP. That's what we're using those for. Okay. So, and that is all There's a summary up here. That is all photosynthesis is. Right? You should pay attention to what comes in, what comes out, where this happens, where it doesn't happen. So if we go back to our light reactions and we kind of make a summary of them, we know that the light reactions occur in the thylakoid. And if we just take a second to jump back, these are our thylakoids. And this part of the chloroplast is our stroma. So we definitely see up here, we can label this again. This is inside the thylakoid space. This membrane is the thylakoid membrane. We'll just abbreviate it as membrane. And then down here is the stroma. So notice what comes in, what goes out, what do we need, what are we going to get. If we make our summary then, the whole point of the light reaction is that we're making a couple of things. We are going to make ATP. We'll kind of keep that in our color codes here. And that's going to go right to the Calvin cycle. We're going to make our electron carrier, and we're going to fill it with our nice little electrons so that we can use those use that energy that's in those electrons to power the electron transport chain and move those hydrogens across the membrane. We're going to need or use, in this case, water. And when we do that, we are going to also then have as a byproduct our oxygen. Let's just label that as our byproduct. So that's the first half of photosynthesis, and the second half are the dark reactions, or the Calvin cycle. And this part occurs in the stroma. It's like that nice little liquid area. Let's put a couple of lines down here so we can figure out what we need and what we use. So in this case, it uses ATP. And I'm just going to put over on the side here that it actually uses 9 ATP. That way it kind of reminds us that we're going to need to use both cyclic and non-cyclic photophosphorylation so that we get enough of the ATP. The second thing it uses is NADPH. Notice we made both of these up in the light reactions. Now we're using them during the dark reactions. It makes our G3P, which is our sugar molecule. Eventually that'll become glucose. And in order for this to work as well, we need carbon dioxide. We can use the same color as we did for water. We'll just use a darker color. We actually need 3 of those for this to work. And that, in a nutshell, is pretty much a walkthrough of all of the reactions, starting from all the way at the beginning with photosystem 2, moving through photosystem 1, making ATP and NADPH, and then going through to the Calvin cycle. Once we go to the Calvin cycle, we fix the carbon, we make sugar, we regenerate our RUBP, and now we have our sugar. Any questions? Please stop by and ask me. Thank you.